So we're going to take a look at the gastrointestinal system here, looking at uh, just as an overview here in terms of digestion, absorption, and the removal of food materials from the alimentary canal. We'll get more specific into each of the individual components here within each of the other presentations. So we have two principal functions here within the system. The two principal functions are going to be to digest materials and then to absorb the nutrients that come about from the digestion of the materials. In doing these two processes, the system is going to be able to provide sensory information as to what materials are coming in so as to initiate the neuroendocrine response for postprandial, that is after meal initiations for fuel utilization as well as metabolism. The digestion, because of the chemical reactions being exothermic, will be able to provide energy to the body to allow for additional heat generation within thermogenesis. The other thing it's going to be involved with is it's going to be involved with overall regulation of energetics for the system based off of this neuroendocrine response that we get from the liver as well as the pancreas in terms of its overall regulations, along with signals coming from adipocytes as well as from the gastrointestinal system itself. So what do we talk about when we talk about digestion? What we're actually talking about is we're talking about the mechanical and chemical breakdown of food stocks so that we're able to get the chemical components of the food stocks into the body itself. Now we have to remember that the alimentary canal, even though it's inside the body, is not necessarily inside the body. It's an open tube that runs from the oral opening to the anal sphincter. And so anything that's in there has to get broken down into its chemical components so that it can go about being absorbed through the villi and the microvilli of the alimentary canal. If it doesn't get broken down into its monomer components, sometimes into a dimer component, it will not be able to be absorbed, with the exception of things that will come in through myoceles of lipids. It's a combination of mastication, mechanical digestion within the oral opening by the teeth. Once you have swallowed, the mechanical breakdown of food stops. Everything else within the digestive process is going to be chemical. The only way to differentiate between the chemical and the mechanical breakdown is the ability to break large food stocks into smaller food stocks. Whereas with chemical breakdown, what we're actually doing is we're actually getting the polymers and the dimers down into their individual dimers and monomers for absorption principles. So the teeth, based off of their anatomical orientations, will go about breaking down the food stocks into larger components, and those larger components will then be able to chemically digested through the rest of the alimentary canal. So let's talk about the digestion of absorption as it goes through the alimentary canal. So we'll start with the mechanical digestion that's going to occur within the oral opening. This is going to take place through mastication. Also, we're going to start the initiation of chemical digestion through interactions of the food materials with saliva. We're going to initiate the peristaltic reflex through swallowing. We're also going to have the initiation of the neuroendocrine response due to the signals coming from the gustatory centers, the taste buds, with the hypothalamus, allowing for the initiation of the neuroendocrine response. That's going to establish both enzymes that are going to come into play later on within the alimentary canal, as well as what's going to happen in terms of postprandial, that after meal response that's going to allow us to deposit all of the monomers that we've been absorbing into the tissues that we need to absorb those materials into for normal homeostasis. So what is saliva? And what's its overall function? So saliva is going to be made up of water, amylase, lipases, immunoglobulins, a whole bunch of immune cells, which includes some lymphocytes. In the study packet, I've given you a large table of materials that you will see in saliva, and you should be aware of those materials. Those materials are going to function to do three particular things. Number one is they're going to produce a lubricated bolus of food. This lubricated bolus of food will allow for it to more easily move through the esophagus and into the stomach, and what's referred to as esophageal motility. It's also going to allow for easier mastication by the teeth. It's going to initiate chemical breakdown, particularly of carbohydrates and lipids, through what's referred to as salivary amylase, which will start breaking down large chain carbohydrates, as well as lingual lipase, which will start to break down small lipids, not necessarily large triglycerides, but smaller free fatty acids. It's also going to initiate any immunological response that we might have to any type of food pathogen. This immunological response can be part of the triggers for food allergies for those people who have types of allergies to distinct types of foods, by initiation of mast cell response and basophil response to release the complement proteins, including serotonin and histamine. So if we look at the alimentary canal in terms of its overall function, we have to look back and remember its anatomy. And so what you remember is that the alimentary canal, even though it's inside the body, is not really inside the body. It's an open tube. It's an open tube that's set up in such a way so that it will protect the body from anything that happens to be within the food materials itself. So in order for food materials to get into the body, it has to be mechanically broken up into smaller components that allow for the enzymes to chemically break apart the food material itself into its monomer and dimer units. We usually do not absorb the polymers within food materials. We typically are going to be absorbing the monomers and the dimers, and more importantly, the monomers within that, which is why the alimentary canal itself is broken up into an 8-meter segment of 3 distinct regions. The first region is all about getting the food bolus in and initiating the chemical reactions to get it into its polymer components. And so that'll be the upper tract, which is going to include the esophagus and the stomach. We then have the area that's going to start continue the chemical digestions down into the monomer and dimer units, and then allow for absorption of either the stuff we put into the alimentary canal or the materials that we are attempting to absorb through digestion, and that's within the small intestinal tract. Notice the small intestinal tract is the largest component of the alimentary canal. We then get to the large intestinal component, and the large intestinal component is going to be involved with a compaction of materials and the last chance for absorption of anything that happens to be within the food materials remaining within the alimentary canal. Regardless of the height of the individual, the alimentary canal will average about 8 meters. Now, very short people will have slightly shorter alimentary canals. Very tall people will have slightly larger and longer alimentary canals. But the adult alimentary canal will be about 8 meters in length, and that's necessary to allow for enough distance for digestion and then absorption of all of the nutrients that we're going to be consuming within the food materials that we're eating. This 8 meters actually gets extended based off the anatomy within the lumen itself by the villi and the microvilli within the intestines and within the small intestine, and to a lesser extent within the large intestine. So what's going to be involved with the chemical digestion of the materials? We're going to have saliva and salivary enzymes, which can be involved. We're going to have gastric juices, which will be involved. We'll have bile and pancreatic juices, and then we'll have small intestinal secretions. We usually generate somewhere between 3 and 4 times as much fluid to digest the food materials as what we actually consume in the foodstuffs. So let's take a look at this overall digestion and absorption in terms of the volumes of materials. So we're going to have somewhere between 1.2 and 1.5 liters of materials being consumed, plus about a kilogram, 800 grams of materials being consumed in terms of mass. These numbers are going to change based off of dietary patterns. We get about 1.5 liters worth of saliva being produced due to the amount of material that we're going to be consuming. These numbers will change based off of increase or decrease of the amount being consumed. Based off of the amount being consumed, we'll have about 2 liters of gastric juices, which are highly acidic, predominantly hydrochloric acid, and about 2 liters combined of bile and pancreatic juices, which will be produced, which are highly alkalinic, predominantly bicarbonate in terms of the amounts. Now, if you notice, the volume of gastric juices is equivalent to the volume of bile and pancreatic juices, and that's necessary so that we can neutralize the total amount of material that's coming in to the duodenum from the stomach. We have to do this so as to protect the intestinal mucosa as well as the tissues of the intestines from the acidity of the hydrochloric acid in terms of both its pH as well as its molarity. Now, some of the digestion that is going to take place is going to necessitate the exposure to acids. So even though we're attempting to neutralize completely, we may not completely neutralize all of the materials coming in based off of what materials are being consumed. Protein digestion is necessitated off of exposure to acids, and particularly the enzymatic reactions that we see with pepsin. And so any type of digestion that's going to involve pepsin needs to have the acidic condition in order to initiate pepsin digestion. We're going to get another about 1.5 liters of materials coming in from the intestines through digestive enzyme being secreted or mucus being secreted along the brush border of the villi and microvilli of the intestines, which gives us a total of around 9 liters of material coming in. Now, unless we're going to try to somehow get that 9 liters intravenously infused into us or somehow absorb the 9 liters through additional consumption of liquid, we have to get that 9 liters back. Of that 9 liters, 8.5 liters will be absorbed within the small intestine. About 350 milliliters to about 500 milliliters will be absorbed within the large intestine based off of hydration status of the individual, which tells me that we get somewhere around 8.9 liters of materials being reabsorbed or absorbed within the intestines itself, which means that we're only losing about 100 milliliters of liquid and about 50 grams of material within the defecation reflex. Now, the defecated or fecal matter that is actually being released is much larger than what is being indicated in the diagram. And that has to do with other factors that are going to be released within the fecal matter, which we'll talk about later on.